So welcome everyone. I would like to welcome you to our Let's Talk About NETs interview series, where today we will be talking about the global phase 3 clinical trial COMPOSE with friend and longtime colleague, Professor Michael. Before we start, I would first like to acknowledge the traditional owners of the land to which I join today, the Bunurong people of the Kulin Nation, and pay my respects to their elders past, present and emerging. We have an exciting interview as part of our series, as we don't very often have a global phase 3 clinical trial available to NET patients here in Australia. The principal investigator at Peter MacCallum Cancer Centre of the COMPOSE study is Professor Michael, a medical oncologist who is the trial and medical lead of the Upper Gastrointestinal Service and co-chair of the Neuroendocrine Tumour Unit at Peter Mac, which is an ENETS Centre of Excellence. Welcome, Michael.
Thank you, Simone.
So, Michael, can you tell us about this trial? And for those who don't know what peptide receptor radionuclide therapy is, a bit of a brief description about that also.
Okay. So on the majority of neuroendocrine tumours, regardless of their site, they have what's called hormone receptors on their surface. And think of them as like a lock of a door, for example. And we know that these locks or receptors is where the somatostatin drugs such as sandostatin, octreotide or lanreotide, for example, bind to the surface of those NET cells and cause those NET cells to either stop growing, decrease in size or decrease hormone production. But we can also use those locks to our advantage because we can actually bind the hormones to a radioactive atom, such as lutetium. And what that does is these NET cells bind the octreotide or lanreotide, and attached to that is a radioactive atom. The cell gobbles up the radioactive atom and therefore causes like a little nuclear explosion within the cell and kills the cell off. And that's called peptide receptor radionuclide therapy, or PRT. And we've been doing PRT for patients with NETs for the last 20 years or so. And it's also obviously run across all the major NET centres across the world. And we've seen very good prolonged tumour control, whether it's stabilisation of disease or whether it's a decrease in the size of the tumours for up to 3 or more years in patients with NETs. And we now have what's called a randomised phase 3 trial called the NET-1 trial, which randomised patients with grade 1 NETs to PRT versus higher dose of octreotide if they had progressed on standard dose octreotide, and found that PRT did provide a better tumour control, much better tumour control than the control arm. So that's become now a universal standard across the world, and it's been funded by the Americans and the Europeans, and hopefully we'll get funding for that in Australia. But we're still doing that as part of our standard therapy in Australia. But that only accommodates those patients with the low-grade NETs, the grade 1s and the low-grade 2 NETs. What about those patients with the higher grade 2 NETs or the low-grade 3 NETs? These are the more aggressive tumours which tend to grow very, very quickly, and we've often previously had treated with chemotherapy. And at Peter Mac, as well as some European centres, have been treating these more aggressive tumours with PRT, either as first-line treatment or patients that have had prior chemotherapy. And we often see in these patients that when we do a GAT8 scan, which I know majority of your NET patients do have, that their disease, especially if they're well differentiated, grade 2, grade 3, the GAT8 scan shows all their sites of disease up, and therefore tells us that we can actually target these disease with the radioactive therapy. And we have seen very, very good responses and disease control compared to, say, chemotherapy alone or drugs like everolimus, for example. Probably not as long a control as, say, compared to those patients with lower grade disease, but again, much better than standard chemotherapy, for example. So we initially put up a submission to one of the companies that makes PRT about doing a trial in grade 3 NETs, and they subsequently took on the idea and developed the idea. So now we have the COMPOSE phase 3 trial. And the COMPOSE phase 3 trial, it's a randomized phase 3 trial, being that we're randomizing patients to either PRT alone or to the best standard care, subject to the doctor's own opinion at the different centres. And the standard of care would be either chemotherapy, such as oxaliplatin 5-FU called Folfox, or capecitabine temozolomide, or everolimus. And these would represent standard regimens for this type of disease. And so patients either have 6 cycles of PRT, subject to their response, versus ongoing chemotherapy or everolimus. And it's for patients with high grade 2 disease or low grade 3 disease. They may have had 1 prior chemotherapy or prior octreotide or lanreotide therapy and show disease progression. And it's for patients whose disease is almost entirely Avid on the GAT8 scan. And if some patients for this trial also need to have an FDG PET scan, which shows more aggressive disease. And most of those FDG Avid lesions also need to be GAT8 Avid. So we're basically targeting all sites of disease with the PRT.
Fantastic. And I think you mentioned that there. And can I just, for the, for the audience and for us, can you explain what Avid means?
Avid means that it basically shows up on the PET scan. So in other words, when the tumor spots have the octreotide receptors on their surface, and we can actually show that on the PET scans. So it just means a level of uptake on the PET scan.
Like many of our patients say, they light up like a Christmas tree.
That's right, exactly right.
And you mentioned then about they may have had another treatment before this, because obviously we're always a little bit concerned about inclusion and exclusion criteria, and we always are like our patients to be seen quickly with regards to trials so that they don't miss out. So they can have had a treatment. That's
right. So they could have had prior chemotherapy and have shown evidence of disease progression on that chemotherapy. And that was really because there's such differences in practice treating this more aggressive NET group of patients. And so therefore there was some reluctance by the committee designing this trial, which myself was part of, and so was Grace Kong at Peter Mac. And there was some reluctance about doing this as a first-line treatment for patients, even though we have done that at Peter Mac. So therefore, we allowed patients who had prior chemotherapy to become part of the trial entry criteria.
Absolutely, because we know many of our patients may have been diagnosed outside of an area of a centre of excellence,
That's right,
like Peter Mac, this trial also will be opening at Royal North Shore, another centre of excellence in New South Wales. But we know, our patients get diagnosed elsewhere, so they are, and because of the aggressiveness of these higher grade tumours, they are put on treatments pretty quickly. So we want then to ensure that they are able to be referred on as soon as possible, and hence videos like this hopefully can raise the awareness about how they can do that. And I guess that was one of the other questions. So how many patients are you thinking that you would like to recruit, or you're required to recruit?
We're required to recruit 5 patients. We'd like to get up to 8 patients or more. It's competitive recruitment over about a 2-year period. They've opened the trial across many centres in the US and in Europe as well, up to about 50 centres from last count, I think. So they're really trying to get as many patients onto the trial so we can answer this very important question as quickly as possible.
Absolutely, and get this treatment available to patients as quickly as possible, and also into guidelines, so that for, again, patients that are not being seen near centres of excellence, that they are going to be then referred on to places that have PRT available. And so I guess that's the other thing, is can patients, or how do patients get referred to you, and can they travel from interstate?
Yes, they can travel from interstate. The patients should be referred by their treating oncologists, which can provide the appropriate background to the patient's past history and prior therapy. And we would, at Peter MacCallum, we would prioritise NET patients quite quickly in terms of be seen as quickly as possible, especially if they're trial patients. They can be seen from interstate. they will need to travel from interstate for their treatments, unfortunately. We did discuss with the trial designers teletrial model, but no one's ready for that at this stage, unfortunately.
No.
So the companies will provide funding for patients to travel from interstate.
Which is great because, again, this is a smaller subset of our patients, and that's actually, you know, so whilst they're high grade, they can be, it's GEP-NET and lung, or just GEP-NET?
GEP-NET.
Just GEP-NET.
Yep.
So those gastroenteric pancreatic
Right.
neuroendocrine tumours. And if a patient wants to travel, or even for a patient who's referred to Peter Mac or Royal North Shore, it's an outpatient trial, so they don't have to stay overnight.
Exactly. Yes,
Yeah.
all the treatments, including PRT and the standard chemotherapy of Valimansums, are all outpatient treatments. So there's no need to stay overnight at all.
No, and I think, look, then they may have, and everything's so well organised, they would then have their scans. And interestingly, you know, this is where it does differ from the other sites from around the globe, where they are inpatients, and we're really lucky to have it as an outpatient service here in Australia. So what do you think? I mean, Michael, this is, whilst again, we are always encouraged by trials that are really focusing on those higher grade unmet need patients, these tumours do grow quicker than the grade 1s and grade 2s. You know, what do you think with the future of sort of the results of these trials? Whilst that's great because we might be able to get reimbursed product, but could we be starting to look also at combination trials as well?
I think that's an area of active research for our NET patients. We have been combining chemotherapy, whether it's capecitabine or capecitabine temozolomide, in PRT for patients for a prolonged period of time. And I think the evidence is mixed about whether we should be or should not be. I think it really comes down to very careful patient selection, and think about it in the high-risk patients. And we certainly do that as routine. And there's now more trials looking at careful selection of high-risk patients where we would add chemotherapy. We're certainly at Peter Mac, we're running a trial called the PARLONET trial, which Grace Kong is leading, and we're adding a drug called a PARP inhibitor, because we know that the way that radiation works is it damages the patient, the DNA or the genes within the NET cell, and the NET cell can't repair that gene and dies. Some NET cells do repair the genes and continue to live on. And so with this PARP inhibitor, what it does is stops that DNA being repaired, therefore increases the damage of the DNA by the radiation from PRT. So we're actually now having what's called a phase 1 trial looking at different doses of PARP inhibitor in combination with PRT. And there's also other studies looking at combining other biological agents to PRT, and including immunotherapy as well. And there's certain discussion within international groups to try and develop trials of immunotherapy with PRT also.
And that's the thing, we know, and because of the long history of 20 plus years with treating neuroendocrine tumours, that we can have patients who respond so well, but then yes, they grow back, or they're stubborn tumours. They like to you know, keep on growing and come back, and so being able to break that DNA cycle, that's incredible. So I think that's a great snapshot of what is a really, really important trial for a very important patient group, and cannot thank you enough, Professor Michael, for everything that you do, and for being part of our interview series.
My pleasure. Thank you for the invitation.
Thank you.